from UCL.
Is this, plug this on here. Sorry, I'm going to write down. Brilliant. Hi, morning. Thanks, Chris, and thanks so much to the organisers for inviting me to present and for organising this event, which I've had a great time so far, and I think I'm looking forward to the rest of today. Okay, so I have a really broad title here, Muscle mNIRS in Healthcare and Observational Cohort Studies. And just to give some background, probably a lot of you will know these things, but just to set the scene, skeletal muscle is important for physical function, for metabolic homeostasis, and for thermogenesis. And muscle function declines in the presence of many diseases and also just through the healthy aging process. And mNIRS can be used to non-invasively assess several really key features of muscle physiology, including vasodilatory capacity and oxidative respiratory capacity for performance. And muscle mNIRS has been used in healthcare research to understand disease mechanisms and to track the response to interventions. However, despite sort of potential utility in clinical healthcare, mNIRS techniques have not really been translated into clinical tools. So my aims for today really are threefold. First of all, to describe two mNIRS measurement techniques and provide examples where they've been used in healthcare research. And secondly, to describe mNIRS application in two observational cohort studies. So this is a little niche, and this is what I've done, and I'm excited to share with you. And then finally, I'll just highlight some strengths and limitations of mNIRS, particularly in the context of mNIRS as a clinical tool. I think these limitations are probably across the board, so hopefully applicable to everybody. So mNIRS can be used to assess vasodilatory capacity. We talked a little about this yesterday in Kevin McCauley's fantastic presentation. Vasodilatory capacity can be measured by quantifying reactive hyperemia following an arterial occlusion. And this is called post-occlusive reactive hyperemia, or PORH. And to do this using mNIRS, you need to position the device downstream of an arterial occlusion. Usually they're 5-minute arterial occlusions. In some studies, they've used 3 minutes. And then reactive hyperemia is quantified following the release of the cuff. So you can see on the, you see that? No, hold on. So you can see in this nice schematic here on the right-hand side, you can see baseline. So these are just the changes that you might see in your oxy-HB signal or your TSI signal during the ischemic phase and then during the reperfusion. So this is baseline, this is just rest, and then there's desaturation during ischemia until the point where you release the cuff at the bottom here, and then you get this rapid reperfusion. And I think in this schematic, the recovery following the reperfusion is slightly accentuated. So sometimes that does plateau a little and then come back down, but in my experience, it always recovers. And the mechanism of this is that there's a drop of pressure inside the small arteries, and you get a reduction in circumferential wall stress, and then myogenic tone is withdrawn, and you get vasodilation of the small blood vessels. And there's various factors released from the endothelium, nitric oxide, adenosine, etc., that contribute to that vasodilation. And we know that a blunted reactive hyperemia, not just with mNIRS, but measured with other measurement devices as well, other techniques, ultrasound, venous occlusion, plethysmography. A blunted reactive hyperemia indicates impaired vasodilation, and this is a predictor of future cardiovascular morbidity. So mNIRS has been used to detect a blunted post-occlusive reactive hyperemia in the presence of a whole range of conditions. And this includes peripheral arterial disease, coronary heart disease, metabolic syndrome, sepsis, respiratory disorders, and the list kind of goes on. It's also been used to compare older and younger adults. And in this figure, which I've borrowed from a paper in 2015, the authors looked at the number of cardiovascular risk factors that an individual had. That's on the x-axis. And then each of these y-axes is a different parameter that you can extract from the reactive hyperemia. So this one at the bottom, for example, I think hopefully you can see the text, but this one at the bottom is the area under the curve of the oxy-HP signal. And you can see that as the number of risk factors increases, there's a reduction in reactive hyperemia. Okay, and then another paper in 2017 showed the difference between young and old adults. So we've got here the young, this is an example trace, young folk in an example young person in black, and then an example elderly person in the red. And then on the right, again, you can see several different metrics which are extracted from either the desaturation slope or the reperfusion. And you can see that these are all much lower in the older folk compared to the younger. But what I thought was really interesting in this paper is that the authors also tried to adjust for the desaturation, to adjust the reperfusion for the desaturation. And they did this, they did a 3-minute occlusion in the younger folk where because of the increased desaturation rate, they dropped to a similar level as the elderly people, even though they'd done a 5-minute occlusion in the older folks. And they did this, they actually attenuated some of the differences between the old and the young. So I think this is really interesting, both from a research perspective, the physiology, but also from a methods perspective, because it highlights this real advantage of mNIRS in that we're able to look at the differences in desaturation locally during the test, so in real time, and potentially adjust for those. Okay, so we can also use mNIRS to assess oxidative respiratory performance or capacity. And this measurement, like Kevin Macaulay told us yesterday, this involves a short period of muscle contractions or electrical stimulation, and then transient arterial occlusions during the recovery phase. So you can see an example here. This is just a resistance band, which we did plantar flexion against. And we have the mNIRS device on the calf here. We've tried a few different protocols. So in this example, we used a 2-second contraction every 2 seconds for 5 minutes, actually. So quite a long protocol. And we did that to try and align with what the folk were doing in MRI, because we were trying to match these up. And yesterday, Kevin said that these measurements do align with phosphocreatine payback, and we were trying to do that as well in the calf. But we've also done, you can do just a short 10 or 12-second contraction phase. Okay, so then what you would do with each arterial occlusion, you would plot that against time. You'd fit a mono-exponential curve through these points and then derive the time constant, which, like I said, is aligned with phosphocreatine recovery. And in fact, that's the theory behind this measurement, is that PCR payback is all the oxidative processes. And so you're capturing oxidative capacity through this measure. And this has been done again in a whole host of different disorders to describe reduced oxidative performance in the presence of a whole range of conditions. So recently, there's been work done looking in people with long COVID. And we've done some work which Alex Jameson's going to present in the next session. And there's also other groups that have done mNIRS work in the presence and absence of long COVID. So it's also been described in heart failure and COPD, in spinal cord injury, and then again recently in response to statin treatment. We also touched on this yesterday, so apologies for all the duplication, but I thought this was really interesting. This is a paper that came out earlier this year, published by Terence Ryan and his team. And they looked at the time constant in people before and after statin treatment. So we've known for quite a long time that statins, while they are fantastic at reducing atherosclerotic disease risk, they do, or they can come with side effects of muscle soreness and muscle weakness. And so this group showed in quite a small study, well, it'd be perfectly sized for Kevin, actually, because it was 8 participants. But anyway, they showed that after statin treatment, sorry, there was a prolonged time constant. So that's impaired oxidative performance. I'm going to change direction now and talk about observational cohort studies. So this is something I'm really interested in and have done a lot of work in observational research. So just in case you're sitting there wondering what an observational cohort study is, these are studies usually quite large, set up to observe a group of individuals. And they're not necessarily a clinical population. Often the aims are to address questions related to aging, healthy aging, disease aging. And data can be captured using just questionnaires or via research study visits or home visits. So where a participant would come into the research lab and they'd have, or research clinic, we could call it, and they'd have blood samples, a whole range of biomedical measurements. And like I said, these studies can be quite large. And they can also involve repeat measures throughout either the life course or one phase of the life course. And so you can see immediately why mNIRS is a really attractive tool to use in these kind of studies, because it's cost effective, it's non-invasive, and it's a portable way to assess muscle function. So the first study I want to talk about is the SABRE study. SABRE stands for Southall and Brent Revisited. And this is a cohort study which was set up in the mid-1980s. And it was set up to investigate health disparities by ethnicity. So at the time, researchers at the time knew that there were higher rates of cardiovascular disease and type 2 diabetes in first-generation migrants, South Asian, from India and also from the Caribbean. And so they set the study up to try and understand why this was, so that we could, provide better healthcare for everyone. So participants were either European or first generation South Asians or African Caribbean adults. And in 2014, so fast forward 30 years, the SABRE participants attended their 3rd research clinic visit. And at that time, they had a mean age of 74. And part of the visit involved an exercise test where we also put a mNIRS device on the calf. And that was in the context of a whole load of other clinical measurements. So they had ultrasound and they had DEXA, all of these kinds of things. But what we found with the mNIRS device is that on average, there was a greater decline in TSI in the South Asians and the African Caribbeans compared to their European counterparts. And in this table, you can see in just under 600 folk there, the mean and the 95% confidence intervals for each group. And you can see different models on the y-axis. So these are just the covariates that I included in my model. So these differences are adjusted for age, sex, and VO2, which we measured during the exercise test as well. And you can see the differences here. And as we adjust for more covariates, we lose some power. So we're no longer able to assign statistical significance to these effects, but you can see that the effect sizes don't actually change that much. So we interpreted this greater reduction in TSI as a reduced ability to deliver or to appropriately redistribute oxygenated blood to meet the demands in the muscle. And this may, an increase, we could also have an increase in extraction, which would compensate for the reduced delivery, and that might also contribute to our TSI signal or our TSI differences. And I think that's quite important to highlight because still with mNIRS or with continuous wave mNIRS, we're not able to pull those two things apart. So you don't know whether the change that you're seeing, unless you apply an occlusion, you don't know whether the change is due to extraction increase or a failure to deliver. So this interpretation was also in line with other measures we did in these study participants, which suggested that there's reduced cardiovascular function in the South Asians and in the African Caribbeans. And the final study that I want to talk about is the Avon longitudinal study of parents and children, or ALSPAC. So this is a longitudinal birth cohort, which was established in the early 90s in Avon, which is a region in southwest England. In this study, pregnant women were recruited. So these mothers who were recruited and their babies, who subsequently became the index participants, these guys were followed with serial research visits throughout their whole life. So every so many years, we followed, or the team followed them up. And research visits really included a wide range of different biomedical measurements, a whole lot of self-reported data, and also physical activity assessments. And the participants have actually just finished their Focus at 30 research visits. So they're now aged 34. And we started seeing them when they were just approaching, or just over 30, in fact. And the pandemic kind of set us back a little bit. So at Focus 30, the visit included a cardiopulmonary exercise test. Oh, and we also put the mNIRS device on during the test. So almost 2,000 people have done this test now. And because the study just finished, I don't have any results to show you. But I wanted to mention it because I'm just about to start processing those data. And I think there could be some, and also the data will become available eventually. So I think this could be a really interesting thing to talk about, different ways that we can process those data and really understand in a healthy or relatively healthy population who have been very well phenotyped, what some of those measurements might mean. So really delighted to talk about that afterwards. I'll just finish with some strengths and limitations of muscle mNIRS, particularly related to its translation into a clinical tool. There's loads of conditions where vasodilatory capacity or oxidative respiratory performance or mitochondrial dysfunction is negatively impacted. And I think there's definitely merit in developing this or developing more clinical tools for assessing those muscle features. mNIRS is non-invasive and it's really cost effective. So I think it's fantastic. It has big advantages to, for example, a biopsy. And again, Kevin said this yesterday, but you would really want to know that you needed a biopsy before you had one. So if you could do a measurement that suggested there may be deficits in the mitochondria, and therefore let's do a biopsy to really understand the detail, I think that's fantastic. It's cost effective compared to MRI. I don't know if anyone here has ever tried to do phosphorus imaging. It's horribly expensive. So yeah, fantastic if you can use mNIRS. The signals detected from the muscle microvasculature, which I think is a big advantage, especially within the reactive hyperemia measure, because often when people look at reactive hyperemia, they can use tools, venous occlusion plethysmography is the famous one, but you're just getting the whole change in the lower limb. You're not really specifying where the signal's coming from. And the same with laser Doppler, you're really only looking at the skin, you're not looking right into the muscle. And there's good temporal resolution. So we've heard about high frequency sampling a lot this morning already. That's amazing. These devices are being more developed more to allow us to do that. And I think there's scope for multi-channel devices as well, so that we could screen the whole muscle. You could screen different places along a whole muscle and really understand what's going on across the whole muscle, rather than just picking one particular point to measure from. The devices can also be made portable and wearable, as we just saw. And this is really applicable, not just in sports science, but also in clinical healthcare, with trying to remotely monitor patients. I think there's definitely scope for that. So a few limitations and barriers to clinical application. There's definitely a lack of robust clinical thresholds. And there's limited consensus on the methods that we use, like the standard procedures that we use, and also on reporting. So what's, first of all, the best practice for correction for adipose tissue? And then which parameters do we report? You saw earlier on a slide with a whole range of different parameters that we can report from reactive hyperemia, but which ones? And I think that really needs to be clear. It can cause a lot of confusion within patients and also within clinicians. So maybe we could address this with clearer protocol papers, additional work assessing precision and accuracy. There has been work done looking at repeatability of both of those measurements that I've discussed today, and also looking at accuracy, comparing it with you know, the MRI measure and the biopsy measure, but maybe we need to do a bit more of that. The data processing steps are often involve custom written scripts. So again, I think we could address this quite simply with consensus papers and maybe code sharing and potentially future meetings like this one where we talk a bit more about that. There's also outstanding technical limitations such as the impact of adipose tissue thickness and the contribution of myoglobin. And I think there's also poor awareness in clinicians. So I think, I don't know, maybe it's not our job, but maybe we do need to promote what we're doing a little better within healthcare research, healthcare clinic. Okay, so in summary, muscle mNIRS has been widely used in healthcare research. It has great potential in observational cohort studies and as a clinical tool. There's clearly methodological issues that we should aim to resolve, and particularly if we're going to have motivation for mNIRS to be used in a clinical setting.